Yeah. Was das jetzt ein Power? All right. So the next presentation is a proceedings presentation delivered by Lai Du, and it's entitled Identifying Progressive Imaging Genetic Patterns via Multitask SCCA, a longitudinal study of the ADNI cohort.
Good afternoon, everyone. I'm being here to present our works about identifying progressive imaging genetic patterns via multitask SCCA, a longitudinal study of the ADNI cohort. Now let me first introduce our background and motivation. Sorry. In previous studies, if we want to know which gene or genetic variation to be associated with the disease, what we used to do is first we ask the doctor or an expert to make a decision about this person if he is normal or abnormal. And then we got his sequencing data, and then we conduct some statistical analysis or some machine learning techniques such as regression to identify if a gene or genetic variation is correlated with this disease. But we ask a question. A doctor or an expert makes the decision based on his own knowledge, and each doctor has different knowledge about the disease. If the doctor makes a wrong decision, or the normal abnormal was determined wrongly, so this will misleading our understanding about the genetic findings. So we ask if we can use some continuous variable to describe the disease, unlike the discrete description of the disease such as normal, abnormal. Then the answer is yes. For example, let us take the brain disease such as Alzheimer's as an example. We can use the MRI scanner to image the brain, and then we can get the continuous description of this brain, like the brain's volume, surface, or its densities. These measurements are continuous, and then it will be a better measurement than the discrete measurements. So our works is the imaging genetics. Its goal is to investigate the role of the genetic variation in disordered brain function using neuroimaging biomarkers as phenotypes. The traditional imaging genetics conducts like this. On the left side, the data matrix loads the genetic data, such as the SNPs, and each subject has thousands of numbers of SNPs. And on the right side, there are his imaging biomarkers, such as brain areas. For each brain area, we denote it as an ROI. Then for these two data matrices from the same population, we can conduct a multivariate analysis between them and find out which SNPs are correlated with which ROIs. One point must to be pointed out is that sparse learning usually used to make the feature selection on both sides to select the relevant two SNPs and ROIs to be correlated with the AD at the same time. But this raises some problems to us because we know that we got disease gradually over time, not suddenly. This means the disease progresses over time. It's not remain stationary. On the top, here shows 3 brains. The first one shows a health brain. In the middle is a mild Alzheimer's. On the right side, it shows a severe Alzheimer's brain. Now let us look at the hippocampus area. We can see that the volume of the hippocampus area changes from the health to the serve AD. It becomes smaller and smaller. This means the hippocampus changes small and small from the normal to abnormal. The figure on the bottom also shows 5 other biomarkers of the imaging. We can see that all imaging markers changes gradually from normal to the dementia. So this tells us the disease changes gradually over time, not suddenly. So the question is, the brain disorder progresses over time, and the brain cerebral declines differently. First, the different brain areas declines differently, and even one brain area declines differently at different stages of the brain of the disease. And the existing methods only can use one imaging modality at one time point. So it ignores the longitudinal information embarrassed in the imaging data. So now let us look at our solution, the temporal multitask SCCA. Our model works like this. On the left side, it also loads the genetic data. But on the right side, there's a cube data set. One dimension corresponds to subjects. One dimension corresponds to ROIs. And the other dimension corresponds to the time points. So we have a cube data set to load the imaging biomarkers longitudinally. So you combine this imaging data and the genetic data together. We use the multitask learning framework to combine them. And in addition, we also use some sparse learning techniques. On the genetic side, we not only consider the LD block information, we first use the G21 norm to constrain those SNPs from the same LD block and across multiple time points. We constrain them together to make a stable feature selection. For each individual SNPs, we use the L21 norm to constrain them across multiple time points. This can help us select SNPs that play roles during all time points. And the L1 norm is used to help us select SNPs that only effective at one time point, not all. On the imaging side, we also use the temporal constraints, but it's slightly different from the constraints on the genetic side. Here we can see that we use a FP21 norm. This norm introduces smoothness between 2 successive time points between one ROI. Because for our brain, the ROI changes gradually. This means at 2 successive time points, it may remain stable. So this helps us to select the ROIs print these characters. And the L21 norm also helps us to select ROIs that remain stable during all time points. And the L1 norm helps us select ROIs that only effective at one time point. So put these together, we have our objective. The first term is the multitask CCA loss function plus the 2 norm, omega u and omega v. This is the temporal constraints we just introduced. Our model has 3 advantages. First, gene data is associated with each longitudinal imaging data separately. Second, it learns multiple SCCA tasks jointly, and each task is for each time point. It does not require one single canonical week to be correlated with all longitudinal imaging data. Then we find that our model is biconvex in U and V. So we first fix V to solve U. After that, we fix U to solve V. And these two steps repeated till the algorithm converge. Now let us look at the experiment results. First, the data source. We use both simulation data and real data. The simulation data, the first three simulation data simulates a problem that with different noise. This means the data 1, 2, 3, they have the same ground truth but different noise. The fourth data simulates a large imaging genetic problem. The real imaging genetic data are from the ADNI database. Here we have 125 healthy control, 192 MCI, and 91 AD. We use the genetic data from chromosome 91 near the ApoE gene. And the imaging data are structural MRI scans across 2 years. The baseline 6 months, 12 months, and 24 months. We segment the brains into 90 hours without the cerebral area. This figure shows the results between our method and the two benchmarks, the multi-view SCCA and the TGSCCA. The correlation coefficients shows that on the first data set, we see that all methods works poorly when the noise is high. As the noise goes lower and lower, all their performance are improved. On the fourth data set, we see that all method performs acceptable. We also found that our method and the TGSCCA performs better than the multi-view CCA. This means both the TGSCCA and our method has a temporal constraint. This may be a good constraint in the longitudinal studies. This figure shows the feature selection results. The first row shows ground truth, followed by 3 rows correspond to 3 methods. Our method's results are at the bottom. We can see that our method can not only identify those active variables at a specific time point, but also identify those active variables shared by all time points. This figure shows the results from the real data. The testing results shows that our method outperforms both benchmarks about the correlation coefficients. This means our method has improved by multivariate correlation coefficients. And this figure shows the identified of the SNPs. Both the multi-view and TGSCCA can only find out the time-consistent SNPs. This means they only can find the SNPs that play roles during all time points. They cannot find out the time-dependent SNPs. But for our method, we can find both the time-consistent and the time-dependent SNPs are found out at the same time. This figure shows identified imaging QTs. The results is the same on the SNPs. Both benchmarks can only find out the time-consistent imaging QTs, while our method can find out both. Here are the details about the identified SNPs. The left shows the time-consistent SNPs, and the time-dependent SNPs are shown on the right. We found that most of the SNPs identified by our method have been previously reported in other studies. This verifies the variable of our method. And this shows the details of the identified imaging QTs. And we found that our method found out most AD-related imaging QTs. It's interesting that the right angular area, it has been reported to be an aging-associated cognitive decline area, and our method found out it to be time consistent. This is interesting. Now let us make a conclusion. We propose a new temporal multitask SCCA. It has improved by multivariant association ability. It selects both time-dependent and time-consistent features at the same time. Thank you for your attention. Any questions?
Any questions? So I would have one question.
Okay.
I was wondering how important the sampling rate for the neuroimaging input is in order to find these these SNPs that induce temporal variation. I mean, you showed this one example where you had, I think, 4 measurements across 2 years.
Yeah.
Can you elaborate on how much you expect to improve performance if you were to increase or lower the sampling rate?
Yeah. We only have the data across 2 years in this study. But if we have more time points about the imaging data, we can use them, test them to verify the performance. Yeah.
Did you evaluate how performance was affected if you were only to use parts of your temporally resolved imaging data? If you were to only include 2 time points instead of the 4? Did you try this out, and how did this affect performance if you did?
Oh, no, yeah.
Okay.
Sorry.
All right. Are there any more questions? I don't see anybody running to the mics. All right, so in this case, let's thank Lai again.
Thank you.